if you have any questions, I'll be happy to answer them.
In regards to, is there any risk of engraftment to the engraftment with actinium being given or Actimab being given, and then 10 days later you're doing the allotransplant?
You're asking a good question. You want to answer that?
Yeah, sure. So the answer is no, not the way that we'll do it. We've done the same kind of preparatory work with the Actimab program, a different program that we have. And basically what we've done is look very carefully at the time that it takes for the radiation that's available in the body to come down to levels that can't cause any harm to the graft. So we've done exactly the same thing in preparing for this trial, is take the PK data we have and basically make sure that the time that we give, and basically 14 days later, is the sort of short answer to your question. You don't have to worry about that because there's no more radioactivity. But certainly you can't give the cells immediately after giving a radioactive material that's aimed at myeloid cells. Exactly right. But it's quite manageable.
Yeah. So I said it's probably wrong saying 7 days. I said it's more going to be...
In this case, it's 14, because we've done the work.
Yeah.
In terms of the spironolactone that was mentioned in the last set of slides, I was wondering if that could be addressed at this point.
Spironolactone.
Yeah, so it's an interesting question because it turns out that the radioactive bismuth potentially can accumulate in renal cells, and in the original animal experiments done, where actually the animal did not have CD33 that was recognized by the antibody persisted for much longer. And bismuth did end up in some cells in the renal cortex, and there was some renal toxicity. So basically, in experiments to look at that, they originally started off with diuretics using Lasix, and ended up using spironolactone as an alternative and seemed to find some benefit from spironolactone. The actual mechanism isn't really understood, and whether it's anything more than just simple diuresis. And also, I have to note that we haven't seen any renal toxicity in our actinium program in patients to date. And so whether this is related to the fact that the animals didn't have CD33 recognized by the antibody, and so there was great persistence of this material, is still a little bit unclear. But we're not seeing that clinically. Nevertheless, we decided to put in spironolactone and use it just as a preemptive measure.
So the NRC, Nuclear Regulatory Commission, as you know, regulates this kind of thing. I just wonder what exposed patient, what precautions, if any, are required by the NRC with regard to body fluids, nursing personnel, rooms, et cetera, et cetera?
So clearly we pay a lot of attention to that. One of the wonderful things about alpha particle therapy with actinium is that alpha particles are stopped by a piece of paper. They're stopped by your skin. So there's no radiation being emitted by people who receive this therapy. And therefore, they can get the therapy as an outpatient, and they can go to the mall, wherever they want to go. However, your point is well taken. There do need to be proper nursing procedures to make sure that nurses administering or people administering aren't in contact with bodily fluids from the patient, because they actually can potentially contain some radioactive material, particularly early on. But otherwise, there's nothing special that needs to be done, for instance, when the patient goes home. And that is one of the real benefits of using an alpha particle therapy.
And that's true in every state. There's no local regulations that affect this?
Well, actually, many of the regulations are state by state. Some states don't have their own regulations, use national regulations. But the answer is yes. And none of this is related to regulation.
I have a somewhat tangential question. In this low CD33 expressing disorders, is there any work on inducing CD33 expression in either plasma cells or?
That's a very interesting question. The only work that I know of is that it turns out that apparently there's some effect from anti-CD45 antibodies that can raise somewhat CD33 levels. It's a potential avenue for the future, but we're not aware of other ways to do so, but it certainly is one way to potentially make this treatment more viable to a larger multiple myeloma population.
Mark, can you speak as to why CLAG-M has been chosen for the AML study?
It's basically investigator-initiated. I mean, I think there's certainly potential to use actinium and actinium-labeled antibodies with other salvage chemotherapy regimens. So no, it was really convenience as much as anything else. In other words, there's no reason not to use it with other regimens.
Mark, if we could also give credit to the Baylor people who started the CD33 program in myeloma.
Yeah, yes, actually, that's actually where the multiple myeloma study started, and much of the, it was started as an investigator-initiated trial there, and actually we've now brought it in-house because we want to expand the number of sites and move the trial faster. But I think that's exactly right. There was a number of different ideas that have come to us from others. The CLAG-M idea, the multiple myeloma idea, and frankly the MDS idea from Dr. Rubos as well. These are ideas that people have looked at this with us and said, well, how can we take advantage of the actual strengths of this drug, and where can we use it?